Hey, this is Richard Jacobs, and I want to tell you about one of my favorite podcasts right now. It's called Product Hunt Radio. Each week, Ryan Hoover and Abadesi from the Product Hunt team interview the founders, investors, and makers that are shaping the future of tech. They've grilled the New York Times' Taylor Lorenz on the future of social media and Andreessen Horowitz's Scott Cooper on the future of venture capital. Every Wednesday, they dive into how tech is shaping our future, from remote working to niche online communities to virtual reality. It's an eye-opening take on an industry transforming our daily lives, but rarely in the most transparent way. Go ahead and listen and subscribe to Product Hunt Radio on Apple Podcasts, Overcast, or in your favorite podcast app. You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you. Hello, this is Richard Jacobs with the Future Tech and Future Tech Health podcast. I have Eduard Willms. He's a postdoctoral researcher at Trobe University in Melbourne, Australia. We're going to be talking about EVs, extracellular vesicles, also known as exosomes, and it'll be a very interesting talk. So, Eduard, thanks for coming.
Yeah, thank you for the invitation. It's great to be here.
Yeah, what got you interested in exosomes? How did they even come across your radar?
I think that this is something that already happened during one of my first research internships in my undergraduate, so my bachelor's, when I was studying pharmacy. And I got the opportunity to do an internship for 6 months at a department at the University of Utrecht with Raymond Schiffelers. And he basically introduced me to one of his postdocs working there. We started doing some basic experiments and getting used to lab skills working with exosomes. So this was cell culture and all kinds of different techniques which are commonly used in the lab, but we were specifically looking at and trying to isolate exosomes as I knew them back then, and extracellular vesicles as people have been calling them now.
Right. So in what context are you studying EVs?
So obviously if we look at EVs are vesicles that communicate messages between cells. I'm looking at it as a cellular communication mechanism. So cells communicate with each other through mechanisms like hormones and neurotransmitters and other types of interactions. And extracellular vesicles are one of these recently discovered methods of communication. And then what I'm particularly interested in is what we've started to call extracellular vesicle heterogeneity. So this is basically looking at what kinds of different types of vesicles are secreted by cells. And this has become very relevant, and I think it's come up a couple of times in the podcast that you've recorded on extracellular vesicles, where we have this idea that cells secrete a lot of extracellular vesicles, and they're all in the bloodstream and in the body and circulating and influencing other cells. And it sounds like this is a very unregulated system, but I don't think nature has intended it to be like that. So I think there is some level of specificity in terms of how messages are communicated between cells and what vesicles travel to what cell and what vesicles are taken up by cells and what functions they carry out. And I think one of the levels of specificity arises from the fact that cells release very specific types of vesicles. So I'm interested in dissecting what kind of types of vesicles are released by cells and what kind of functions they have.
Well, how do you capture the vesicles given off by a cell? Can you have it in culture with like a net around it somehow, where you can scoop up all the EVs it puts out?
No, so basically it's kind of a reduced approach, I would say, like a reductionist approach. So obviously we try to look at one cell line. So you culture these cells, and then these vesicles are secreted into the liquid basically that you grow these cells in. And then you can take the liquid and you can do a couple of purification steps, which involves centrifugation. And then what we've been using, classically people have been using ultracentrifugation. So if you look at the literature, those are techniques which were originally used. So spinning the liquid that you take from the cells at very high speeds, and then you're able to sediment these vesicles and then study them. But we've been using a technique which is called size exclusion chromatography. So basically using the difference in size between larger vesicles and the smaller proteins which are secreted by the cell as well. You can separate them from these proteins and have a relatively pure preparation of vesicles to study.
Well, I would think that if you put two cells together that are the same cell type, you get one kind of vesicles. If you put two different cell types together, you get another one. And then, I guess if you put a microbe in the mix, you may see communication between someone's somatic cell and a microbe as well, or a virus, or, there's many different permutations you could do, I guess, initially just to see major differences in what the cells are putting out, or, change the pH with given cells in solution. You can do I would think hundreds of different permutations to see how things change just to start with.
Exactly. Yeah, this is people have been trying all kinds of different things to influence the vesicles that are secreted by cells. But it's challenging, obviously, due to their small size, first of all. I think that makes it very challenging to study them. And then also, like I said, because there's most likely all kinds of different types of vesicles released, and isolating these and having pure populations is a very challenging thing to do, which makes it quite complicated, even to study them in a setting of one cell communicating with so cell A communicating with cell B. Even that can be quite tricky.
Well, why? Why is it so hard to separate one cell in one medium and one in another?
I think what specifically makes it difficult is we don't really know how many types obviously are released by a cell, and the mechanisms which are used for their secretion by different cells are similar. So if you look at the composition of the vesicles, you probably find shared content. So this would be proteins that are shared between the vesicles. So just like cells are composed of certain building blocks, these vesicles will be composed of certain building blocks. But within that, there are most likely differences. And because you're looking at it.
Why couldn't you have, for instance, one cell in a medium and profile what's in the medium? Yeah. And then, feed it, have it sit there for a few hours, and then profile again how the medium's changed. And then maybe again, raise the temperature and see how it changes.
Yeah,
and maybe
sure.
Start with one. Why not that?
You can definitely do that. And I think there's definitely people trying these things.
Well, what have you seen in the literature? What have people tried where they've published results that they whispered them to you, on the phone or something? And what have you seen people do, and what have they learned so far?
So one of the things that people have been looking into, I guess, is how these, and this is also something that I'm interested in, is how these vesicles play a role in cancer, and then in particular the metastasis or the spreading of cancer. So this is something we can talk about, which has been quite well studied for vesicles. So in terms
tell me what's been found there.
Yeah, so this is so if you look at cancer, we obviously know that cancer spreads, and so this is what they call metastasis. And one of the kind of unknowns there is how is it how do cancer cells spread? And why is it specifically that, for instance, a breast cancer would spread to the lymph node or to another distant organ? And what's one of the processes which is involved in this is what they call a pre-metastatic niche formation. So it turns out that before the cancer is actually spreading, there is already kind of a niche formation going on in, for instance, the lymph node. So that the environment of the lymph node is already changed to make it more susceptible for the cancer to grow there. So this is this niche, so a pre-metastatic niche, which is formed before the metastasis actually occurs. And what has been shown is that extracellular vesicles that are released by cancer cells are able to induce the formation of this pre-metastatic niche.
What does the formation look like? Are there lymph cells that? Are there cancerous cells literally sitting in the lymph? What does this niche look like?
No, so this is the vesicles, not physically the cancer cell. This is the vesicles secreted by the cell. They're traveling to, for instance, the lymph node, and they are influencing immune cells in that environment, or they can, for instance, induce angiogenesis, so the formation of blood vessels once the cancer cells arrive at that distant site. So this is the kind of processes that have been shown to be influenced by extracellular vesicles.
All right. So what's interesting then is there must be a reporting back of, is it ready? Is it time now to metastasize? Otherwise, why would they start feathering the nest, for the cancer cell in the first place?
Yeah. Interesting point. Yeah.
So what's the mechanism? But, perhaps EVs, they're taken into cells. I mean, what are the range of things can they do? Can they change the gene expression of that cell? Can they change the outer membrane proteins that are produced and open channels or closed channels? I mean, can they change the functionality of the cell completely? What can they do?
Well,
I think cause that cell to send back EVs saying yes, successful migration.
Yeah, I don't think anybody has actually looked at that. As far as I'm aware, nobody has really looked at when is there a signal that actually goes back? Is there a communication in the reverse in that sense? So that's actually quite an interesting point. I'm not sure.
Yeah, I mean, again, I tell you I've said this to other people. When I think about a cell, how does it has to, okay, well, I know it's going to get metaphysical, but it has to know that it is itself, and it would communicate, I would guess, in different ways with, again, if I'm a hepatocyte, I'll know, okay, I'm this kind of cell. And if I'm going to communicate with other hepatocytes, I would package specific EVs for them. But if I'm going to communicate with some distant organ, how do I even know how to communicate with that other organ cells? How would I know that? So I have to have essentially a language that tells me what to package up and where and how to target, distant parts of the body. But again, I'd have to get feedback that it worked somehow.
Good point.
And then all the incoming stuff, as a cell, how do I know what to let in or what not to let in? And when I do let something in, how do I open up the box and see what's inside and say, okay, this is this and this is, how do I get the package and read the from message? It's from my neighbor, the bone cell, and here's what the message says. I mean, that's all communication and discernment.
Yeah, this is exactly the point that I guess what I want to raise as well. We see all this and we think this is how it's happening, but there should be a certain level of specificity. And like I said, I think one of the levels of specificity might arise by a certain signature that is present on a given EV. And we haven't been able to show that properly yet. So what kind of vesicle is able to really enter a cell and deliver its messages, and where is there differences in where certain EVs deliver their message to, which is obviously highly relevant, because if you look at, just to change the conversation a bit, if we look at how people are thinking of using EVs, we all want to use these extracellular vesicles. I think this is a big part of the literature and a big part of the discussion that is going on is how can we use these extracellular vesicles to deliver certain therapeutics or drugs into the cells. And here it becomes obviously very relevant to know which signature on the vesicle, or which type of extracellular vesicle is able to do that trick, because that's the kind of vesicle you want to package your therapeutic in. Well, and we don't have an answer to that yet.
Yeah, one way is to look at it that way, but the other way is, what is the primary way by which a cell takes in a vesicle? Like, where is that discernment happening? It sounds like it's happening at the outer membrane, the lipid bilayer, right? That's where it's happening?
Yeah, I think there's probably surface interactions going on which allow a cell to decide if this vesicle is even allowed into the cell, yes or no. And then,
so what if you were to profile? Is another huge job, but what if people were to profile, ideally, all the cell types, let's say in the human body, their outer membranes? What does it look like? And then we could make this kind of cell, what other kinds of cell could it potentially communicate with, or what would it need to be able to make a vesicle? What properties would it have in order to enter into these other kinds of cells? Perhaps then we would see a pattern like, the membranes of these kinds of cells are highly selective and very little will get in, while these other membranes, they'll take anything in,
Yeah, I think it's a very interesting approach, and there are some people that have looked at uptake of vesicles between different cell lines. So again, studying in a more reductionist approach, so looking at one cell line, if you feed this cell line vesicles, how many are taken up? Looking at another cell line, feed the same amount of vesicles, how many are taken up? But I think dissecting from that information what actually dictates the yes or no signal is very difficult. And I think it's especially difficult with extracellular vesicles, because you're not just looking at one protein. You're looking at a composition of proteins. You're looking at a lipid bilayer with different lipids. You're looking at sugar molecules or glycans which are on the surface. And then there's also the content, so that there's nucleic acids inside the vesicle which might have effects on where this vesicle ends up, or what trick it does. So I think that the approach you mentioned is obviously things that we in the field are thinking about as well, but it's very complicated.
Well, maybe you should take a very special circumstance, like right before an egg is fertilized by sperm. I bet you the sperm are spitting out EVs and the egg is spitting out its own. And there's, again, it's just pure speculation, but
more reduced approach and a more reduced setting in that sense. So the human body is already so many different cells that it's quite difficult to study.
I think that may be a good approach, because that's a very rare and unique event.
Yeah.
So that under those conditions, you may be able to much faster elucidate what's going on, because I bet you there's a communication happening. I don't think it's just, sperm number 47 is like just able to push its way into the egg faster and stronger. I bet you there's communication and discernment on what will be let in.
Yeah, there most likely is. And I think the approach you say, like go studying in a more simplified environment, might actually be what we have to do to start understanding this. And I think, or studying it, this is something that we're interested in, studying at a more like a single cell level, and even a single vesicle level. So these are kind of techniques and approaches that we're starting to get to grips with, because the techniques that are used in our field are getting better as well. So we're getting down to resolutions which allow us to look at these vesicles and look at their composition, and even single molecules on the vesicles. So I think we will be able to do these kind of things, but I haven't seen anybody really doing it yet. So.
Well, you want to do this. So what experiments are you cooking up in your brain? What do you think will be the best way to go at this, if you're willing to reveal it?
Yeah, sure. No, I can talk a bit about the work that I've already done and that I'm doing at the moment. So, like I said, I'm interested in this heterogeneity. So looking at what kind of different subpopulations, we call them, so what different types of vesicles are released by cells. And I think you've probably seen some of the original literature, and a lot of people there talk about a couple of major populations that we've been studying. So you mentioned exosomes already. So it turns out exosomes is one of these populations that people have been studying over the last couple of years. And the exosomes have a very specific biogenesis, like a formation pathway in the cell, which is known. Like we know some of the proteins that are involved, and we know some of the processes that happen in the cell before this vesicle gets secreted and is called an exosome. And then you've probably also seen things we call microvesicles. So people have been using these as this term for the vesicles that are released directly from the cell membrane. And another type of vesicle you hear about a lot is called apoptotic body. So this is an extracellular vesicle that is released when a cell undergoes apoptosis. So maybe communicating to the rest of the environment, hey, I'm undergoing apoptosis. This is what happened. This has happened to me, and here's some information that might be useful to you. We don't really know. But what I'm then interested in is taking these extracellular vesicles, and then I use an approach with size exclusion chromatography, where I'm able to separate all the EVs that are secreted by cells and have more distinct size ranges within that. So I can get different populations of vesicles with different ranges of sizes. And then I'm looking at these different populations and characterizing them for their protein content. So what is unique about them, and looking at their functionality. And we are then looking, in this case, I'm using this in a setting of ovarian cancer. So looking at how ovarian cancer is able to spread to the abdomen. So that's one of the projects I'm working on.
So what have you found so far? Like, what are some of the thoughts that have come into your mind? What are the observations?
Yeah, so like I've said a couple of times, I think not all vesicles will have the same function. So there will be different types of vesicles released, which have different functions. And one of the things we're looking at is how do these vesicles play a role in ovarian cancer spreading? And what we see there is, so what people have shown is that you have in this case, in ovarian cancer, you have the primary tumor in the ovary, and the metastasis or the spreading is able to happen directly through the abdomen, so via what they call the peritoneal fluid. So there is a direct link there, not via the bloodstream, but directly into the abdomen. And what happens is the primary tumor sheds cells, so the cells become released from the primary tumor. They travel through the abdomen, and they have to establish there in order to form a secondary tumor. So in order to establish a new tumor in the peritoneum. And we have observed, and other people have shown, that extracellular vesicles are able to support adhesion of the free-floating ovarian cancer cells to the abdominal wall in simplified way. And I'm looking at how
do they do that? Do the EVs stick to the abdominal wall, or do they
exactly.
The nature of it?
Yeah, so we think a part of it is probably using it as an adhesive scaffold. And and this is also, if we look at the proteomic content of the vesicles, you can really see that there is high enrichment of adhesion molecules. And these properties of the vesicles have actually already been exploited in a mouse model, so where researchers have looked at can we use these EVs to influence where the ovarian cancer spreads. And so what they did there is they made a scaffold which they coated with the extracellular vesicles. So they isolated extracellular vesicles from, in this case, also ovarian cancer, coated those extracellular vesicles on this scaffold. Then they surgically implanted that scaffold into the abdomen of mice and injected ovarian cancer cells and looked at the spreading of the ovarian cancer and the survival of these animals. And what they saw is that these mice, they lived much longer. And if they looked at where the ovarian cancer cells were after sacrificing the animals and looking at how the spreading had happened, they could see that the ovarian cancer cells had mainly migrated to this scaffold, which was coated with extracellular vesicles. So they call this approach, they call it a pre-metastatic niche mimic. So it's like mimicking the pre-metastatic niche formation. And this basically shows you that extracellular vesicles play some kind of role in mediating where these cancer cells end up. So that's quite interesting.
Was there have you tried to do that? Have you tried to coat, well, not you, but the researchers, did they try to use a certain amount of scaffolding? Like, was there a threshold below which the cells wouldn't adhere? There was, quote unquote, too little scaffolding?
I'm sure that they probably done like different ranges of concentrations. I'm not sure. But yeah, this is something we're going to try with it. Like I said, we've isolated different subpopulations of extracellular vesicles from ovarian cancer. So we're working together with this lab, which is based in Spain.
Here's why that may be important. If you only put up a little bit of scaffolding and the cells don't adhere, that means there is definitely a communication back and a sensing of when is it, quote unquote, enough to go ahead and now send out cells to adhere, I would think. Because if it doesn't vary by the amount of scaffolding, then no. But if it does, that might give you a major clue.
Yeah. Well, this is
like quorum sensing in a way, but a different kind of quorum sensing,
Yeah. I like this. I like the idea that you say about looking at, how does it actually signal back? But, we actually, this signaling might not occur through extracellular vesicles. So this is also something to take into account. Like, we're trying to link all these things of cellular communication up to one specific mechanism, but obviously there's different mechanisms which are at play in the cellular environment. So this is also something to take into account, I think.
Do the EVs decay if they're out of the cell for a period of time? Like, do they change? So
you mean they're
alive? Are they? I mean, like, what happens to them? How long do they stay before they fall apart?
Yeah, good question. So there's stability. So in actually, like, I don't know how there are some studies out there which have used isolated EVs. So they purified EVs and then used certain labels, injected them into mice and looked at their biodistribution. So where do these vesicles go? How long are they in the circulation for? And I think I'm not 100% sure about the details, but it's like most nanoparticles, they're not in the circulation for that long. Like, it's quite, none of them stick around for that long. So there's certain organs in which they accumulate, which I think is liver and kidney. So the main organs that are actually involved in the excretion. But otherwise, they're not actually around for that long.
But when they do adhere, though, and they become a scaffolding, like, do they preserve their form, or do they turn into a semi-scar tissue? Like, what happens to them? Do they, if a lot of them are there as a scaffolding, do they attract the immune cells? I mean, do we know what the fate of them are once they've made a scaffolding?
No, we don't actually know that. No, I don't know what exactly happens, and if there's maybe also different cells which adhere to them, which is very likely. But there might be some, because they are ovarian cancer-derived vesicles, there might be some level of specificity towards ovarian cancer cells. But we haven't looked at this, and I'm not sure.
I mean, there's 8 million questions. Like, tumors are heterogeneous, right? So
as well.
Are they? Is the tumor? I mean, which cells? I don't think they're randomly shedding cells, maybe. But let's say a tumor has, 100 different kinds of, gene expression variations in the cells. Mm-hmm. What kind of cells, are there any predominant kinds that end up going to form the metastasis once the scaffolding's there? Is there any preferentiality or not?
Yeah, I think there's I don't know in terms of is this actual, like, there's likely ovarian cancer cells which are more prone to spreading. So I've seen some information about this in the literature, but I'm actually not sure if these. You would assume that these are also the cells which end up spreading and adhering to your scaffold. So that's something which would be very interesting to check.
No, but even if you just confined yourself to the ovarian cancer cells themselves, again, the tumors themselves are heterogeneous. So to call something an ovarian cancer cell, there's many different forms of that. It's not just one type of cell. They're not monolithic, So is there any preferential subtypes of ovarian cancer cells that would stick to the scaffolding? Do they It's just so many questions. Like,
yeah,
it's crazy.
There's definitely a lot of questions, which keeps us busy, I guess. Lots of research to be done.
A lot, yeah. Huh. So, all right, well, I've been asking you all the questions. I mean, any insights that have jumped out at you since you've begun this work? Anything that really, like, blows your mind, or any directions you see to go in the near term that you're working on?
That I'm working on myself. Oh my.
Even in partnership, but specifically, what are you going to be doing?
Like I said, I recently moved lab. So this is my first postdoctoral research position, and moved from the UK to Australia. And the lab that I'm currently working in is very interested in Alzheimer's and neurodegenerative disease. And one of the things that we're trying to do is come up with an early detection mechanism for Alzheimer's or neurodegenerative diseases. And I think there as well, it's the same. We're fishing for a certain signature which could give us a clue about if there is development of Alzheimer's going on. So kind of trying to optimize that by looking at the different types of vesicles that are released, and finding a more sensitive way to actually isolate and look at the specific vesicle which is relevant in that setting. So I think development of a biomarker, which we would call it, so a way to have an early detection and look at, for instance, efficiency of treatments. That's something I'm quite excited about.
Okay. I don't know, what would be a really exciting result for you to achieve in the next few years with your research?
Yeah, I think for me, getting a better insight in the different types of vesicles, and working out which interactions and which protein composition or which signature of a vesicle dictates what a vesicle does. So like I said before, I don't think all of the extracellular vesicles that are secreted by a cell are doing the same thing. So that there must be some kind of something is dictating what they do. And this could arise from the fact that they release different types. Could also actually arise from the fact, the context in which this vesicle is reaches. So it's almost like a nature-nurture discussion. It's like, yes, this vesicle has a certain makeup, but it also depends on the environment it finds itself in. So, for instance, the cell it reaches, or the state of the cell it reaches, on what that vesicle is actually going to do. But for me personally, I think I would really like to find out what is governing this. So what kind of characteristics are dictating what the vesicle does, and then that also will allow us to have a better insight in how to use these vesicles as therapies. Because like I said, not all of these vesicles might be able to enter the environment of the cell. Some of them might not even enter. They might just signal from the outside. So what is unique about the vesicle that goes into the cell? And can we take those characteristics of that specific vesicle and make a I don't know, almost like an artificial vesicle which has those characteristics, and can we then use that vesicle to deliver drugs into cells? I think that's something I would like to contribute to.
Well, how many EVs do cells put out? Do they put out hundreds or thousands or millions? Any sense of the rate and the number?
I don't know. Yeah, I don't know the exact number, but it's from what we see, it's a lot. And I think this is also what has been blowing my mind. I don't think they can all be doing the same thing. I don't think that the nature has intended it to be that messy.
I'm sure they're not all doing the same thing, but
no, yeah.
Again, what does that tell you if a cell puts out 10 different kinds of vesicles?
Yeah.
But it puts out 1 million of this and 5 million of that, and only 45 of these or 2 of these. Have you seen vast differences in the number of EVs, even though they're different types?
Not specifically in that sense. We do observe that there is definitely a release of the more smaller population of vesicles. So there's definitely more of those that a cell secretes. But what is actually dictating that, or if that makes them more or less important than the larger vesicles, we don't know.
Well, sorry, and then you said some bud off from the membrane, but some are created internally. Where are they created? like the endoplasmic reticulum? Like what, in a particular organelle, or like where do they arise?
These are organelles that are involved in that endoplasmatic reticulum, and then there's like the formation of what they call the multivesicular body. So I think this is something that has come up in your previous podcast as well. So it's basically a structure that is formed into which vesicles are formed as what they call intraluminal vesicles. So there's lots of tiny vesicles within that multivesicular body, and then that multivesicular body fuses with the cell membrane, and the vesicles that are released from that multivesicular body are then called exosomes.
And is the cell releasing them in big bunches or one at a time?
I'm not sure. I'm actually I think there's probably multiple multivesicular bodies in a cell at any given time, but how they actually, what the dynamics of release are, I don't know. These are all things which haven't been studied in that much depth, to be honest.
Yeah. Out of all these questions, are there groups looking at all of these, or is there so much to do that, and is it so new that most people are not looking at most of it? Like where do you see the focus, in the EV world? I don't know if if you have your eyes on a lot of the literature, you probably do, but where are the main thrusts in the EV world you see right now?
Well, I guess it depends also on what kind of part of the EV world people are studying. I think, like I said, it's a very interesting field, and also something very interesting to study, because it's not just one protein. It's like a collection of signaling molecules which are in one package. So there's a lot of people, like I said, that are focused on using these vesicles as what they call a drug delivery tool. So it's like, how can we use these vesicles to deliver medication? So there's even a lot of companies looking into this now. And then there's people who are very interested in the underlying, so what we call the biogenesis. So how are these vesicles formed? So this is like, for instance, Clotilde Tari. She's quite a big name, a well-established name in our field, and she's really looked at the biogenesis of exosomes, so really dissecting this process that I just described with the multivesicular body, sorting of vesicles, and the release upon fusion with the plasma membrane. So that's, in that case, that's Paris, so that's France. So there's a lot of people working on this, which makes it, like I say, it's a very exciting field, and it depends on what area of research you're interested in, where the main focus is. Okay.
Well, very good. So Edward, I know all I did was ask like 8 million more questions that could be answered, but hey, that's the nature of it.
No, that's. I mean, it's like I said, it's very interesting to discuss all these things, and I think there's still a lot of unknowns. I think that's what makes this field interesting as well. But it's highly promising, obviously, like you say, cells are releasing lots of these vesicles, and surely there must be a reason why they're doing it. So I think, it will.
There's not any poor souls out there who say it's all random, is there? There probably is.
Well, there probably is, and I think, if you look at this is something that comes up. If you look at, how we thought about these vesicles originally, people are like, oh, this is just cellular debris. So it's just, basically it's the cells putting out their trash, right? And I think still maybe a fraction of the vesicles which are actually released might be trash. Like they might actually not be doing anything. But we can't, like I say, we don't know. Like there is no way at the moment to distinguish between those two, because we haven't been able to really look at like a single vesicle level. So it's like I say, like why is this one specific vesicle able to enter a cell and deliver a message? Why are they all able to do that, or is it just one particular vesicle? And what is unique about that vesicle compared to the one that wasn't able to do that trick? So I think these are the kind of questions which really excite me, and I think are very relevant to answer.
Okay. Well, very good, Edward. I think we're at the end. What's the best way for people to get in contact with you and ask you 10 billion questions, or suggest collaborations, or work with you and stuff, since you're in this field?
Yeah, no, sure. Always happy to answer questions or give people more information. So I think I'm happy to pass on my email, so you can include that in the notes and that come with the podcast, or even LinkedIn. I'm on LinkedIn, so if people want to reach out through that, they're happy to connect over LinkedIn as well.
Well, very good. Well, Edward, thank you for coming. I appreciate it.
Yeah, thank you very much for inviting me.
You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you've enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you.